So we talked about electron transport chain twice, okay, and so we are now talking about electron transport chain occur in the Cristae. Cristae, the folding of the inner membrane, and this is the summary. On this side, it is the summary of the electron transport chain that we talked about twice. I don't like this cram everything, not easy to see. I prefer the previous figure. Is this larger, easier to read. So here is just repeated information I just told you. It doesn't hurt to hear it again. So energy is used to pump hydrogen ion into the intermembrane space. And then hydrogen moves back to the matrix to release and capture to form ATP by ATP synthase. So in other words, the first part here is active transport. So this is active transport. Again, let me emphasize the energy here comes from the high energy electron. Okay, the energy here does not come from ATP. The energy here comes from high energy electron, not ATP. Okay, remember. For this one, it is we should call this facilitated diffusion. Facilitated diffusion. Why is it a facilitated diffusion instead of a simple diffusion? Well, it is a diffusion because the movement of hydrogen ion is moving from high concentration, the intermembrane space, to the low concentration, the matrix. And it does not require energy. But why do we call it facilitated? Because it has to go through a membrane protein called ATP synthase. When it requires a membrane protein for the diffusion, we call it facilitated diffusion. And this process is called chemiosmosis. Chemiosmosis is basically the making of ATP by the ATP synthase, this step here. So as you can see that the organization of the Cristae, the folding of the mitochondria, the inner folding. You can see electron transport chain is here, and this one, the last one, this is the what I call a cytochrome oxidase, because it makes water. We call this water metabolic water. And then you can see that the intermembrane space has a very high hydrogen ion concentration. The matrix has a low hydrogen ion concentration. So what happened? The hydrogen ion will go through the channel provided by ATP synthase. So it's like a diffusion, or it's the same analogy we use in the photosynthesis. It's like waterfall. It's like a waterfall. Ooh. Let me draw it out. So, how many of you have heard about hydroelectricity? So you have a mountain and then you have water. The blue thing is the water. The water goes down the mountain, right? And then you can have a turbine here. The turbine, well, will be propelled by the water. Oops, sorry. So as water go this way, the turbine will go this way. Am I right? In other words, the turbine, the water moves the turbine. As the turbine moves, then you can make electricity. That's the whole purpose of hydroelectricity. So the analogy goes like this. This is the, okay, it is difficult to write. Intermembrane space. Okay. It has a high concentration of hydrogen ion. Here, it is the matrix. Okay, matrix is not the sci-fi movie trilogy. It's not the cyberspace matrix. It is the fluid field inside of the mitochondria. It has low hydrogen ion concentration, low concentration of hydrogen ion. Okay. It is a natural tendency for the molecule to diffuse from high concentration to low concentration area. And this turbine, what will be this turbine? This turbine will be the ATP synthase. ATP synthase. That's how ATP synthase make the energy. So instead of repeating the entire picture here, just like what we did in the last two times, I only talk about this is what we call the chemiosmosis. Chemiosmosis. This is the chemiosmosis. I summarize the chemiosmosis here. ATP production in the mitochondria. Okay, well, do you want me to repeat the third time? Do you get it? It's already 1 hour 30 minutes, so I should move on quickly. So if you don't understand it, feel free to re-watch the two times that I explained that. So let's give you a summary or basic organization of the Cristae. So in the mitochondria, you have the matrix. Matrix is the fluid filled space. And you have the intermembrane space because mitochondria has double membrane. So the outer circle here is the outer membrane. Inner circle is the inner membrane. And then in the inner membrane, intermembrane space, inter means between. intermembrane space, you have high concentration of hydrogen ion. In the matrix, you have the low concentration of hydrogen ion. So in other words, it is a little bit acidic in the intermembrane space. It is a little bit basic in the matrix. Okay, this picture summarizes how many ATP we make in each steps, okay? Okay, in glycolysis, it occurs in the cytoplasm. It makes 2 ATP. Make 2 ATP. Okay. In Krebs cycle, it occurs in the matrix. Okay, it occurs in the matrix. Matrix of the Krebs cycle. Total, okay, total make 2 ATP. Why do I say total? Again, at the end of glycolysis, we make 2 pyruvate. 2 pyruvate become 2 acetyl-CoA. And then each acetyl-CoA goes to 1 citric acid cycle. Therefore, each citric acid cycle produces 1 ATP. Because we have 2 acetyl-CoA, therefore we have 2 citric acid cycle occur simultaneously. Then we have 2 ATP made. Is that clear? Okay, again, pyruvate is a 3-carbon molecule. Acetyl group, the acetyl group in the acetyl-CoA is a 2-carbon molecule. 3 carbon minus 2 carbon, you have 1 carbon. Where is the 1 carbon? The 1 carbon become carbon dioxide. How come it is 2 carbon dioxide instead of 1 carbon dioxide? Because we have 2 acetyl-CoA. So for the citric acid cycle, remember acetyl-CoA has 2 carbon molecule. It is a 2 carbon molecules. And the 2 carbon molecules, when it go through citric acid cycle, the 2 carbon molecule acetyl group, they will become carbon dioxide. So they will become 2 carbon dioxide gas to come out. But because we have 2 citric acid cycle occur simultaneously, we have to multiply by 2, then we have 4 carbon dioxide here. And for glycolysis, we make 2 NADPH. For the intermediate step or the preparatory reaction, we also make 2 NADH. And then for each citric acid cycle, we have 3 NADH. Because we have 2 citric acid cycles occur simultaneously, we have 6 NADH. For each citric acid cycle, we make 1 FADH2. Because we have 2 citric acid cycles occur simultaneously, we have 2 FADH2 made. And then remember, we said that NADH will make 3, each NADH will make 3 ATP. So you get 6 here, 6 here, and then 16 here. Wait, NADH. So each NADH will give you 3 ATP. So 3 times 2 is 6. 3 times 6 is 18. Why is it 16? Oh, weird. This is so weird. It should be 18. Let me see if the math add up correctly. And then this will be the 2. Yeah, each FADH2 will give you 4 ATP. Let's see if we add up together. 16, 16, 12, 12 plus 18, 30. 30 plus 4, 34. let's say if it is 4, 4 times 6, 10, 28, 28, 32. So yeah, this is incorrect. It should be 18 here. I don't know why they call it 16. So you may ask me, okay, why is it 4 or 6? Okay, it really depends on how, which textbook you are using. Okay, some textbook they said that there are 2 types of NADH. There are 2 types. One type of NADH they are used only in glycolysis. The type of NADH used in glycolysis, well, let me see. Okay, I don't remember. Okay, one type of NADH will only give you 2 ATP, but one type of NADH will give you 3 ATP. That's why sometimes it depends on which textbook you use. If the textbook said that it is 2, then you'll be 4. If the textbook said that if the NADH can generate 2 ATP, then you'll get 4. If the textbook said that the NADH can generate 3 ATP, you'll get 6 here. So it is either 32 or 34 ATP from the so-called electron transport chain, and then total. So you add the 2 ATP from glycolysis and 2 ATP from the Krebs cycle. You have a subtotal of 4 ATP, and then you add the 32 or 34 ATP from the electron transport chain. Then total cellular respiration will give you 36 or 38 ATP. So the energy produced is approximately 686 kilocalorie, and they are stored in the glucose and oxygen. That is the total amount of energy stored in the glucose and oxygen. But the product is carbon dioxide and water. It's only 263 kilocalories. What about the rest of the energy? The rest of the energy, okay, the phosphate bond in ATP has a 7.3 kilocalorie of energy. If we take 36 ATP as the total ATP we produce, then all the ATP we have, it has 263 kilocalorie as well. So 263 divided by 686, it is approximately 40%. Remember in previous slide we talked about the efficiency is about 40%. That's the reason. 40% is the 40% of the energy from glucose. So 686 is the total energy from the glucose and oxygen. 263 here, this is the energy from the ATP. The rest of the energy is lost as heat. The question is how much? So you do 686 minus 263. The first 263 is from the product, carbon dioxide and water. And then the second 263 is from ATP. Then you get the number, heat. Okay, I made a mistake here. It's just this part. Okay. So 686 is the total energy from glucose, the reactant. Reactant, okay. 263 is from ATP. The rest, so it's about let me see, let me double check. It's about, oops, let me see, 3, 2, 4, 2, 3. Let's hit. Well, I would say neglect this part. Usually people don't talk about this part. Yeah, usually people just talk about the energy from the reactant, the energy from the ATP, and then that's it. Yeah, forget about the energy content of the product because we don't use them. It's the product. So this is the heat. So this picture is basically a summary of everything we talked about in the last, in today's lecture. It's very long, yes. You need to know the location, Krebs cycle is in the matrix, and then electron transport chain is in the inner membrane or the Cristae of the mitochondria. And then you need to know the number of ATP it makes for each step. You also need to know the total number of ATP. You need to know the input molecule and the output molecule for each steps. So that's about it. Well, let me know if you have any question.